Awesome. So we are basically right on time. So let's get started. Good evening. Thank you so much for joining us for part three of the Winter Wildlife webinar series. The series was something that the Commission started brainstorming about 2 years ago to explore the region's diverse landscape, wildlife, and conservation efforts. And after a lot of positive feedback and interest from last year's programming, we decided to organize another series of four workshops with the help and expertise of our local incredible partners. In January, we received a frankly horrifying but educational close-up on the invasive sea lamprey in Lake Ontario and Great Lakes region. And last month in February, we got to learn more about key bat species in the Tughill, their benefits to the environment, and some of the challenges that these populations are facing. Today's webinar is going to be focused on the American chestnut, which is a species of tree that once dominated the Appalachian corridor, and it had a large native range that even included parts of New York State. So Dakota Matthews is our expert today, and he will be mostly introducing himself, but he is going to be speaking to us. He's a molecular biology lab manager with the American Chestnut Research and Restoration Project at SUNY ESF, and he's going to be guiding us through the past, present, and hopeful future of this tree species that was once pretty typical of the region. Before I hand things off to Dakota, I would like to just make note of a few housekeeping items. For this webinar, attendees are going to be automatically muted and the video feed turned off to conserve bandwidth. If there are any questions, please do feel free to type them into the chat box at the bottom. We're going to be taking questions at the end of the presentation, and Dakota's going to be able to give you some feedback on those. The webinar is going to be recorded and later uploaded to our Commission's YouTube page. And the links to that recording, as well as the new registration for the fourth and final workshop on raptors, will also be sent out in follow-up emails. So with that, I'm going to get off my little soapbox and I'm going to let Dakota do his thing. Dakota, take it away.
Thank you. Hi, my name is Dakota Matthews. I am an upstate New York native. I first started on the American Chestnut project back in 2016 as an undergrad, recently transferred, and then I stayed on to do my master's, which I finished in plant biotechnology in 2020. And then I stayed on again because I like it and have continued on since then as the molecular lab manager here at SUNY ESF. So today I'm going to talk about the American Chestnut Research and Restoration Project here. Going to start with a brief history, talk about what we do, and then where our project is going from there. Thanks for having me. So the American chestnut was once one of the most predominant trees on the eastern U.S. In its dominant range, it was estimated to comprise 1 out of every 4 trees in eastern U.S. forests. And here's a picture of Shenandoah National Park in 1912. And you can see the chestnut trees. They are the ones that look speckled with snow. American chestnut trees flower in July, so the phenomenon was kind of called snow in July. And it was a huge part of eastern U.S. forests up until the mid 1900s. The American chestnut has a variety of forms and traits. On the left here, you can see American chestnut timber type forest canopy competing variety of American chestnut. You can see how big they used to get. These people down here. It was once called the redwoods of the Eastern Coast. And not only did it reach upper stories hundreds of feet, but also there are these nice spreading, almost oak-like patterns to some of the American chestnut morphotypes, and which made it a great park and street tree as well. American chestnut had highly valuable wood product, mainly because it was very rot resistant and easy to work with, nice and lightweight, and you wouldn't have to treat it at all to use it for any outdoor capacity, whether it's fence posts for pasture land, siding for barns, beam barns, all of that stuff. There's also a huge secondary market for American chestnut wood for cabinetry and other types of small wood carving. There was also a huge agricultural aspect to it. farmers would let their fodder or their livestock fodder on American chestnuts early and late fall to fatten up before winter. It's also a great source of gluten-free flours. So we've actually made chestnut beer before, and you can also use it for baking and all of that stuff without having any gluten. There are some medicinal uses to American chestnut as well. There was a study looking at the extract from chestnut leaves that actually inhibited some staph infections, and you can see this cool little apothecary box of dried chestnut leaves back in New York. And then there's also a huge societal and historical aspect to the American chestnut. I'm sure everyone knows chestnuts roasting on an open fire, but also you can't go to a city on the East Coast without finding a chestnut street right next to a maple and an oak street. And so it was a huge part of America. It's also a huge keystone species. We like to say it was useful from bees to bears. It provided a reliable mast crop yearly, as opposed to a oak where they have separate masting years. The American chestnut reliably made tons of chestnuts every single year. And since the demise of the American chestnut, there are estimated at least 5 insects have gone extinct as a result. So what happened to the American chestnut? We introduced a pathogen called chestnut blight or Cryphonectria parasitica, and what that pathogen fungus does is it enters a wound on a tree, and then it begins to colonize that wound and start secreting oxalic acid as a virulence agent, which kills the living tissue. The fungus follows it around the tree and girdles the tree, killing everything above it. Um, this is what Cryphonectria looks like when cultured in the lab. And as you can imagine, if 1 out of every 4 trees where you live just started dying randomly, it caused widespread panic. Here are some headlines from New York City when chestnut trees started to die from this blight and no one really knew what was going on. And as a result, there were just dead stands of American chestnut all across the East Coast. And it kind of looked like a tree graveyard, I would imagine, um, which is a problem not only for the societal and economic value, also the ecological aspect, as well as biomass for forest fires. So here we have a map of the chestnut blight range. In the dark green is the area of predominance. So that's where you would estimate 1 out of every 4 trees was an American chestnut. And then the light green area is where the chestnut could also live and thrive and was found commonly. And around 1876, Japanese chestnut began to be imported into New York harbors, and it was mainly imported as an ornamental and orchard species. But at the time, no one really had an idea of invasive pathogens, so we were just bringing trees in. And then in 1904 was the first description of chestnut blight cankers forming on trees that were lining the walkways of the Bronx Zoo. Then throughout 50 years, the chestnut blight spread throughout the natural range of the American chestnut, killing approximately 4 billion trees. And not only did it spread from the throughout the range of the American chestnut, but also continued on into some other chestnut species, Allegheny chinkapin and Ozark chinkapin, devastating them as well. And why do we still have chestnut blight today, even though American chestnuts are basically functionally extinct? And the reason for that is it can survive on oaks as well. So it causes some small cankers on oaks and it can reproduce on oaks, but it doesn't kill oak trees. And it's honestly hard to notice if you don't know what you're looking for. So how do we still have American chestnuts today? We're lucky enough that American chestnuts have the ability to survive by sprouting from the root collar. The American chestnut, um, because the American chestnut can sprout from the root collar and the chestnut blight is unable to compete with the microbiome in soils, chestnut blight can't kill the root system of American chestnuts. It can just keep killing the top part of the tree, it dies back, and then it resprouts. So we're kind of in a cycle of never getting to full maturity and not able to sexually reproduce, producing the American chestnuts. So at this point, really what's going on is just isolated trees that are surviving off of large stump sprouts, and they're distributed throughout the range. Here you can see that kind of snow in July thing I described from the Shenandoah photo. You can see one chestnut surviving in this whole forest. There are isolated trees all across the range, but they never are able to reproduce, usually because the tree dieback happens before they are able to flower, which is usually between 5 or 10 years, 5 and 10 years. And then also, chestnut trees cannot pollinate themselves. They have to have a counterpart next to them to produce seed. So if you do find a large chestnut tree, which you're able to do, it's often that in the fall, unless there's another one nearby, all of the nuts, when you open them, are going to be shriveled and unpollinated. So, what are some ideas for chestnut restoration? Um, One of the first things that they did in the Bronx when they started noticing these cankers forming on their trees was use a horse-drawn cart to cart out what I would imagine is extremely toxic pesticides, and they applied them to the cankers. And I'm not sure if you can see on your screen, but these guys are wearing no protection and they're just like painting it on a tree. I cannot imagine um, doing that now. There was also, as unfortunately that didn't work. And then there was also a initiative to remove blighted and healthy chestnut trees ahead of the spread of the blight. So that's sort of like creating a barrier to quarantine the trees and hopefully contain the pathogen in one localized area, keeping some alive in a different area. Unfortunately, they didn't know at the time that it also survived on oaks. So you can see in these old pictures, there are some of these oak trees that have almost what they would look like cankers, but it wasn't feasible to clear cut swaths of forest to try to contain a pathogen that they didn't know how it was spreading. There was also another initiative to replace American chestnut trees with timber type Chinese chestnut. This is a good idea at the time, even though Chinese chestnut are more of like an orchard style tree where they don't reach the canopy like American chestnuts do to compete. Um, and unfortunately, they're just not competitive enough in eastern forests to really thrive and replace the niche of the American chestnut. And at the time, they were planted between the late 40s and early 50s, and only a few have persisted to date, so not really um, ecologically relevant. This is probably the coolest one to me. They essentially tried to create Hulk trees by blasting American chestnuts with radiation to the point of basically killing all of the nuts and then stepping it down to see if they could randomly induce mutations to confer blight resistance in these trees. Unfortunately, that did not work, but it was a cool idea and um, would have been unique to see. One of the common ways that is used now in orchard settings and in Europe is through biocontrol. We'll talk about two things. The first one is hypovirulence. Hypovirulence is essentially the inoculation of the fungus itself with a virus that weakens the pathogen. So we're essentially inoculating blight pathogen with a fungal virus that makes it less virulent and not as easily able to kill the tree. This works really well in Europe because there aren't that many strains of Cryphonectria in Europe when compared to America. And hypovirulence has to be applied to an individual canker, and it needs to be transferred from one fungal type to another, and they have to be compatible. If they're not compatible, they won't exchange the virus, and you can apply it, and it doesn't do anything to the blight. So with a less diverse population of Cryphonectria in Europe, it's easier to just apply multiple types of virus or multiple types of fungal strains that are infected with the virus, and one of them should be able to transfer the virus to the virulent strain. In the US, we have so many different types that unfortunately it's not really a uh, feasible strategy um, for restoration purposes. If you're doing this for an orchard style where you just want to produce chestnuts, you can easily use this to maintain mother trees that are not resistant to the blight. Another cool way to uh, individually treat cankers or maintain individual trees in an orchard setting is through soil compresses. So, like I said earlier, the fungal or the Cryphonectria parasitica, the blight fungus, can't compete with soil microbes. So what we do is essentially wrap and create a soil compress or mud packing on an individual canker. We leave it on for a season, and when we take it off, we've actually healed the canker. So you can kind of see what that looks like in the pictures up here to the right. You have a virulent canker that is actively killing the tree. It's all orange, it's spreading. Once you wrap it in wet soil, seal it up, and leave it for a season, you can see where the canker used to be, but it heals up. It kills the fungus, and the tree can recover from that. But again, that is not a feasible strategy for restoration. We can't uh, hire an army of people to just wrap trees in mud. And but it's a great way to maintain individual trees or mother trees. Um, Another strategy that was used was hybridization with compatible species. So American chestnut is endemic to North America, but we also have sexually compatible species, Japanese chestnut, Chinese chestnut, and European chestnut. The Japanese and Chinese chestnuts co-evolved with this fungus, so they are naturally resistant to the chestnut blight. European is much more susceptible, and then American is the most susceptible. So by crossing these species, we could, in hope, maintain American chestnut genes while introducing resistance to the American chestnut population. Unfortunately, you can see here just by this globe diagram, they have been separated for millennia. So the species have diverged so much that you're doing a lot when you're crossing these species. So there can be a lot of effects with hybridization in plants, not just American chestnuts, just hybridization in general. Large scale genome rearrangements, genomic shock, all of these things can happen when you are crossing really diverse different species, even if they are compatible. So it does work in some cases. In some cases, it's not ideal. There are some documented problems with hybridization in chestnut, reduced growth and intermediate traits. So when you cross American chestnut with Chinese, they're not going to be as resistant as Chinese because things are diluted down, and you're not going to get these tall timber types that we're hoping for if we're crossing with an orchard style tree. Um, there are also some issues with male sterility. For instance, when you cross an American with a Chinese, there are cracked bark syndrome where it looks like the tree trunk is exploding. That is done when or that can happen when you cross American and Japanese chestnut. There's also internal kernel breakdown that causes basically the nut as it's forming to just turn into brown goo when you cross a European hybrid with Chinese. But the nice thing about this is even though this can happen, most of these things can be managed, prevented, or bred out of future generations. It's just hard to manage those when you're trying to reintroduce a species to its natural range. You can't go out and manage which trees cross with each other like you could in an orchard setting. And so hybrids may be suitable for restoration in some areas, but for wide scale restoration to the natural range, it might not be the best option if we want to get these nice, tall East Coast redwood like trees back again. So, what we're doing here at SUNY ESF is genetic engineering of the American chestnut. And why that is useful for restoration in these purposes, I'll kind of explain with this book analogy that our old professor, Dr. Powell, initially came up with, which I still love because it made me really understand it when I first knew or first was learning about the project. So if you imagine a Chinese chestnut genome as a book, chestnuts have 30,000 gene pairs. So imagine that as a 30,000 word novel. Hybrids, half of the genes will be Chinese, the other half will be American. So just imagine converting half of that book into Chinese and it's all random. It's going to be hard to understand and it's not going to be the same book. Backcross trees are essentially hybrids that are then crossed with American continually to reduce the amount of Chinese chestnut gene alleles while retaining American traits and hopefully resistance from the Chinese. And they have approximately 1/16th Chinese chestnut genes, or 1/16th chestnut Chinese chestnut words in this book. Excuse me. So that's approximately 10 pages or 1,800 words of this said novel. So we might get in those crosses, make me more resistant, but that also might include other phrases or other gene pairs that make the trees shorter or make the nuts larger and not really staying true to the American phenotype. So if we switch over to genetic engineering, the nice thing about genetic engineering is that we can make a very small targeted change if we know what we want to do. And with that, we can confer or blight resistance without making these wide-scale genomic changes. So instead of randomly inserting Chinese words for English words in this hypothetical book, we would just be adding one or two words to the existing book. And so this little quote here, it was very exciting at that season to roam the then boundless chestnut woods of Lincoln. What we're really doing, instead of converting things, we are just inserting two genes that are represented by two words, blight tolerant chestnut woods of Lincoln. So that is the strategy that Dr. Powell initially decided would might be a better way to go about this in future. So our result and product would be a 100% American chestnut tree with the inclusion of two genes that confer blight tolerance. So now that we have this methodology, why we want to do it, what genes would be candidate genes for resistance in chestnuts? So we took a mechanistic approach to determine what genes we would want to insert to confer resistance. We have to see how the blight fungus actually attacks the tree. And like I said earlier, it enters a wound on the tree and then begins secreting its main virulence agent known as oxalic acid. And that's essentially a very strong acid that the fungus secretes, and it's on the edge of this canker front. So it secretes oxalic acid, it kills the tissue, and then the fungus follows, digesting the dead tissue. So Dr. Powell came across a paper describing a gene called oxalate oxidase. This gene is found in many plants and enzymes, or many plants and fungi, and this enzyme detoxifies oxalic acid into hydrogen peroxide and CO2, both beneficial byproducts for the plant. And how oxalate oxidase works is it is synthesized within the cell and then it's naturally exported to the cell wall where it attaches to the cell wall and actually acts as a first barrier of defense for the oxalic acid. So as the oxalic acid front is approaching the cell wall, these enzymes start to work to detoxify the acid before it can kill the cell. And what's really important about this gene is that it doesn't act as a pesticide. So we're not killing the fungus. There is no cytal activity, and it acts more like a detoxification agent. It just detoxifies what that pathogen is using to kill the tree. And this allows the fungus to survive on the tree, and it also allows uh, the fungus to reproduce, which lessens the selective pressure for the Cryphonectria parasitica, the blight fungus, to evolve to overcome the oxalate oxidase because we're not killing the tree or we're not killing the fungus. And we are also looking at other genes, for example, PGIPs and lactase genes to stack with OXO to fortify resistance, to kind of enhance the effects of oxalate oxidase. So, like I said, oxalate oxidase is a ubiquitous enzyme in plants. You eat it probably every day. This is a list of commonly cultivated food plants that are all contain oxalate oxidases. And so, by introducing this into the American chestnut, and say you wanted to eat chestnut, you're not going to be eating a novel protein that isn't something that you don't already eat every day. And then we also wanted to make sure that um, it's a gene that is commonly found in the natural range of the American chestnut. So these are wild and ornamental plants that are all found within the range of the American chestnut, and even some fungi and bacteria that all have oxalate oxidases as well. So when we introduce this tree, this transgenic tree, we're not going to be introducing a novel enzyme that hasn't been found already in the American chestnut range. So now we have a candidate gene. We have a idea that it could work. How do we actually get that gene into the American chestnut for further testing and to see how feasible it is? The first step is to isolate immature embryos from American chestnut, pollinated American chestnut flowers, and we harvest those nuts before they are fully mature. We bring them into the lab, we sterilize them, and then we open up the nut and extract the immature embryos from the seed. And those are found up in the upper part where this little, almost like Hershey's Kiss, up in the top part of the nut. And while this seems relatively straightforward, it is not, because American chestnuts are not cooperative when it comes to trying to grow them in tissue culture or really work with them in general. They're pretty finicky. So the success rate for this was really low when it was first being tried in our labs. Approximately 1 out of every 1,000 embryos that were extracted were able to produce somatic embryo cultures that we were able to then multiply and maintain. And so once we had some of these established somatic embryos in culture, we began agrobacterium-mediated transformation. And agrobacterium is a natural genetic engineer. What that bacteria does is it infects a plant cell and stitches in genes to that plant cell to start forming crown galls. And if you've ever walked around a park, forest, whatever, and you see a tree that looks like it has just a giant wart on the side of the trunk, those are often agrobacterium galls. And it was formed by agrobacterium editing the tree genome or an individual cell within that tree. So we can manipulate that process by cutting out the genes of the agrobacterium transformation plasmid. We can cut out the genes that the agrobacterium would normally insert that makes the crown gall and instead, or makes the gall. And then we can instead insert our desired genes that we want to have put into the American chestnut. So in this case, it would be oxalate oxidase. So we bathe those embryos in agrobacterium, kill off the bacteria, and then what we have is a American chestnut embryo with possible transformation events. So before we did this with oxalate oxidase, we started with GFP because we wanted to see if we could actually do it first. And so in this picture it's a transformed American chestnut embryo. And each one of these glowing dots is what we call an event. And those glowing dots are individual cells that have been transformed with the green fluorescence protein, or GFP. And you can see that green fluorescence on a GFP scope. So we were able to transform American chestnuts with this. So we moved on to the genes of interest. And we test each one of those events. And the reason for that is Agrobacterium isn't as specific as one would like. Agrobacterium inserts the genes of interest randomly into the genome. It also inserts them a random number of times. So that one individual glowing cell that you saw on event 1 might have the gene of interest on chromosome 3 at this spot. The other glowing dot that represents a different event might be on a different place in the genome. And then some of them might have multiple copies, 2, 3. We've had some that have had 20, 30 copies. So it's important to screen each one to find the optimal conditions, which would be a single individual copy in an area of the genome that is highly expressed. So this is known as the position effect. So we screen all of these transformation events and eventually found some that looked really good. And now we've done all the molecular aspects of this, but we don't actually know if it'll work. So we need to grow up these trees and actually plant them out and do actual field testing to see if we can determine any resistance to the chestnut blight. So we do this via tissue culture. So those individual transformed events are then induced to form shoots in tissue culture, and they grow in these little cubes with everything that they need. And then we start to manipulate them into growing into real trees. We induce root formation. We transfer them from this gel media into soil, put them in a high humidity chamber, let them get used to being real trees as opposed to just growing in gelatin. And then we're able to pot them up and put them into the greenhouses and then eventually the field. So our first initial tests to see if these transgenic American chestnuts were a viable option was to produce all of these clonally. So we just replicated the individual events that had the most potential or promise. We also clonally propagated controls. So American chestnuts that are non-transgenic. We have our transgenic darling chestnuts here. These are all clones of each other. These are all clones of each other. And then the Chinese were also all clones of each other, all from tissue culture. So we grew those up into pots and we did small stem assays, which I'll show you in a little bit, and determined by inoculating them with fungus that we actually have some potential here with darling trees. As you can see, 6 weeks after inoculation, all of the Americans have died. They have resprouted from the root collar down here, so that's what the living tree tissue is from. 3 of the Chinese chestnut controls wilted and died back, while some of them did survive. And then all of the transgenic trees were still surviving 1 year after inoculation. Okay, so we've tested our clones. They have potential disease resistance, but we can't just produce a bunch of clones and distribute them across the range. We want locally adapted trees. We want to conserve genetics by integrating them into existing populations. And we also want local adaptations, like a Georgia tree is going to grow better in the South than a New York tree or a Maine tree, and vice versa. So we want to capture that. So we're not going to just produce thousands of clones and distribute them into the natural range of the American chestnut. So we have to actually start producing these trees and diversifying the genetics of them. So how we did that, this is my old boss, Kathleen. She taught me everything I know, besides Kristen. I think she's on this. She discovered that if you place an American chestnut in a highlight growth chamber simulating peak summer conditions, you can induce an American chestnut or a Chinese chestnut to produce female and male flowers within a year, usually. Compare that to American chestnuts out in the wild or out in the field settings. It usually takes 5 to 10 years for them to be able to start flowering. So instead of waiting for our trees to get 5 to 10 years old and then collecting pollen and doing crosses, we're able to accelerate that process to create new generations of plants and outcross those plants every single year, which was a huge boon for our project. Without that, we would still be working on a way to mass produce these chestnuts. And how we do this is once they're in the highlight chamber for a certain period of time, they'll start producing catkins. They're stressed. And we rub the catkins with a glass microscope slide. The static cling of the glass actually adheres the pollen to the glass slide. We dehydrate the pollen. We store it in a negative 80 degrees Celsius freezer. And it is able to be stored for a significant period of time until the trees out in the field are ready to be pollinated. Then we rehydrate the pollen on the slides, thaw it out, and then we can very accurately apply only the pollen we want to the female flowers out in the field, which allows us to track the genetics of both the male and the female parents, and it allows us to do a lot more detailed record keeping. This is a scanning electron microscope that image that of the pollen anther heads of the American chestnut flowers. So those little yellow balls that we would rub the glass slide along, this is what the inside of those anthers look like. So all of these little footballs are American chestnut pollen, and then all of this is the outside of the anther of the male flower. We also had a very ambitious undergraduate student this year during pollination season do a really unique case study where he tested pollen that we have stored in our negative 80 to determine how pollen age in the negative 80 freezer, how that viability drops off each year that has been stored. So we had pollen that went all the way out to 6 years of storage. And what he did is determine pollen germination rates in lab settings. So inducing pollen to produce pollen tubes and prove that it's still viable. And then we also use the same pollen to then go pollinate trees out in the field to determine successive seed set. And he determined that 4 years out, you still get around 35% pollination germination rate, which is still pretty reasonable for storing something in the freezer for 4 years. So we do these controlled crosses out in the field with the pollen that we produce in the lab, and we then have to test each individual nut to see if it has the gene, because you get half of your genes from your mom, half of your genes from your dad. Trees do the same thing. So what we do is we use a coring needle to take a core of the nut tissue on the side of the nut because we don't want to damage the embryo. We split that core in two and we put it into histochemical solutions. If oxalate oxidase is present in the nut tissue, it'll start converting the oxalic acid that's in the histo solution, and it produces hydrogen peroxide, and that hydrogen peroxide reacts with a component in the solution and starts to produce this black color within about 20 minutes. If it's non-transgenic, it doesn't have the oxalate oxidase, the core just remains a milky white. So we're able to test all of our crosses. This past year, we had 13,000 nuts from our controlled crosses. So we cored every single nut, tested each one, and were able to determine which ones were transgenic and which ones were not to plan for planting. So as we start outcrossing all of these trees and start producing a bunch of seedlings, we actually need to see, does the small stem inoculation data that we got with the clones correlate to the seedlings? So we have been doing extensive small stem assays over the past 10 years on either clones or sexually produced seedlings. And these two green bars here are the transgenic seedling T1 generation, so the first generation from the clones. Those are inoculated the same time as this non-transgenic American chestnut control. And then we also have a resistant control Chinese chestnut. So you can see the transgenic tree canker size significantly smaller than the other two. And you can tell also on these photos, you can see the orange on the two transgenic cankers. You can see it, but it's not progressing. It's not spreading. It's not girdling the tree. It is just coexisting on that tree stem. And then the American completely girdled 29 days post inoculation. And then the Chinese is doing better, but not quite as well. So how these stem inoculations work, you get a pure plug of agar with fungal culture, Cryphonectria parasitica on it. You make a wound on the stem of a tree. You apply that fungal plug with the fungus facing the tree stem. And then you seal it up so it doesn't dry out. And then you watch the canker progress. So that individual was inoculated. You can see the canker here, but it's pretty localized. It's not spreading a bunch. And then this is the full sibling. So these two nuts came from the same cross, just one inherited the gene and one did not. And so that full sibling does not have oxalate oxidase. And within the same period of time that the canker on the transgenic just progressed about, a few millimeters beyond the site, the non-transgenic counterpart was completely girdled at that point. So here's just another example. We have quite a few of these. So this one's pretty unique because we did the same thing, inoculations, full sibs, and within 6 weeks, the entire branch that we inoculated had completely girdled and died off when compared to just this small canker on our transgenic sibling. So we're doing all of these inoculations, but we actually do want to make sure that we're not causing any damage to the fungus. We want them to coexist. We don't want there to be any cytal activity because we don't want selective pressure for them to evolve. So we inoculate some of these trees with the purposes of letting the cankers progress and then re-isolating the fungus from the cankers 6-8 weeks beyond that. So these are fungal plugs that we re-isolated from cankers that were formed by inoculation. So it just shows that the fungus is coexisting with the tree, sort of like how it does with the Chinese chestnut and Japanese chestnut, as opposed to non-transgenic Americans where it just kills the tree. And then this is one last controlled inoculation where you can see the mechanism, whatever the Chinese chestnut resistance comes from, it creates these swollen cankers, these almost blistering swollen cankers, but it doesn't girdle the tree in all cases. And that's essentially what our transgenic trees are doing as well. We're allowing the fungus to live, but we're not allowing it to girdle the tree. So we've been planting these out in the field for years now, crossing, maintaining, or trying to maintain genetic diversity. And we don't inherently want to inoculate all of the trees that we are planting out in the field because we want to see how natural blight infections look on our transgenic trees and their non-transgenic siblings. This is mainly because those stem inoculations are a very harsh test. Not often in natural settings are you going to get a pure plug of pathogen directly injected into a fresh wound on a tree. These are often damage from insects or crooks and branches where the blight enters through the tree. So you can see on this image here, we have some natural canker formations. These are blocky. They don't look uh, necessarily like the inoculations where we do controlled plugs. These this is its non-transgenic full sibling on the right here, and this canker is uh entered through a crook on the tree, so where the branches split, and it is devastating this tree. This branch completely died back, and these other two were on the way at the time of this photo. And then this is just one last photo of these cankers. They look so blocky. This is a transgenic tree, but it's not girdling the tree. Everything above it is still alive. These dead, dying leaves is because I took this picture in the early spring, right after thaw. And yeah, it's a unique canker formation that allows the tree to still pass nutrients from the roots and down from the leaves, but it doesn't look pretty. So, because these are transgenic, we have to go through regulatory petitions through the USDA, the FDA, and the EPA. The USDA because we want to plant it out into the environment. The FDA because we're technically creating a transgenic food source, and the EPA because we are regulated by them for some reason that we still can't quite figure out. Mainly because we interact with a pathogen. If our trees interact with a pathogen, we're regulated by the EPA. We have a petition into the USDA that has been accepted, and our environmental impact statement has been accepted as well. The petition is publicly available to download, and we expect a ruling on that from them sometime in the future. We don't really know at this point. The FDA, we have a biotechnology consultation that review is in progress, and we have a registration and tolerance exemption review in progress with the EPA as well. We've also done several non-target comparison studies. I'm going to run through these relatively quick, mainly because I'm going longer than I intended. We did a tadpole survival study. Our chestnut leaves are going to line vernal pools. If they were once one out of every four trees in the eastern U.S. forests, it's going to comprise a significant portion of vernal pools for tadpole survival. So we did a tadpole survival test in the lab where we grew them up with different leaf types as detritus for them, and and found that there was no real difference in survival or hazard ratio between non-transgenic American chestnuts and transgenic American chestnuts. We actually found that American beech, the tadpoles did not like that. And that's one of the species that replaced the American chestnut after its loss. We also did nutritional analyses. We compared non-transgenic and transgenic nut tissue. In this instance, the fatty acid profiles, we saw basically no difference between transgenic, which are these green bars, and non-transgenic, which are the blue bars. We did see a pretty drastic difference between American chestnuts and European chestnuts, which are these yellow bars here. So there isn't really much of a difference. The transgene doesn't have an effect on nutrition, but chestnut background can. We also did bumblebee and oxo chestnut pollen feeding studies. We made microcolonies of bumblebees and fed them either oxo containing American chestnut pollen or and a high level, a medium level, and then no oxo. We didn't see any difference between survival probability and levels of oxo in the pollen. We did see survival differences between the sources of bumblebees that we got. We've also started long-term ecological research. We have started these demonstration forest plantings in 2019. These mimic different forest and restoration methods, whether it be hybrids, non-transgenics, transgenics, in forest plantings. These three locations are across the eastern U.S., trying to replicate three different locally adapted areas. These will be valuable for environmental interactions, photosynthesis and metabolism, effective pollination distance, natural dispersal, all over a long scale period of time. We've also started doing long-term field testing up here in Syracuse, New York. We have let these trees grow for many years and have been continuously studying how they perform so we can best select which trees we want to move forward when we're allowed to start planting these out. We have some next steps for chestnut beyond our darling trees. We have new transgenic trees that are coming through the pipeline that have OXO on an inducible expression promoter. So all of our darling trees, all the trees I've talked about so far, are expressing oxalate oxidase at high levels all the time. With inducible expression, we essentially have a dimmer switch that controls the gene or OXO expression. And this is sourced from poplar. Excuse me. This poplar promoter is induced when exposed to fungal pathogens as well as when a tree is wounded. So it seems like a good candidate for um, American chestnut transformations. We're also looking into CRISPR applications where we can edit existing genes in the American chestnut that are potential promising genes, but they don't necessarily have high enough expression for us to know if there's any difference. Lactase genes being one of them. That gene essentially helps strengthen cell walls. We're also increasing diversity through breeding and our new GE lines. So we're, like I said, maintaining genetic diversity by outcrossing between non-transgenic mother trees out in the wild and our transgenic pollen. We're also looking into trying to develop, not trying to, we are developing phytophthora-resistant American chestnut trees, which is a huge problem down south, and will be steadily moving further north as climate change warms and uh, makes our winters more mild. Well, I think that is it. I just wanted to talk about how you can help. You can follow us on our ESF Chestnut webpage. There are a bunch of links to our socials, things like that. You can plant chestnuts. Some other trees are very important. Like I said, American chestnuts can't pollinate themselves. So if you have an established orchard or a couple of American chestnut trees, that would allow you to cross-pollinate when these trees become available. And we also would love to hear from you, share your support, all of this stuff. I believe this photo is from one of our trees, non-transgenic mother trees that we planted up with you guys. I think it was last year. I have to go back and check the email, but if anyone can tell me how it's doing, that would be nice. And then I can take questions. Thanks for your time.
Awesome. Thank you, Dakota. Actually, I can tell you where that photo was taken. It was taken in Zoo, New York in Watertown, and I believe some of your folks, along with a number of other people, were there for a nice education session about the darling trees and the research that's been going on for these decades. So that's actually right by the Tug Hill Pavilion in the Zoo, New York. You can see
Awesome.
the regional map. So kind of perfect that you ended up choosing that. Um, and I do see we have a couple questions in the chat. So I'm going to start with the one that was asked first. MJ Dowling asked, my friends in Ohio still buy Buckeye trees, a type of chestnut. Are they resistant to the blight?
So I is that horse chestnut?
It
Buckeyes?
says Buckeye trees. I'm not sure.
Yeah, I'm pretty sure that's just a common name. They are not uh true chestnut species. So they can't cross-pollinate with Chinese, American, European. Um it's sort of like just horse chestnut. Horse chestnut, that's the common name of the tree, but it's not a chestnut tree.
So it really depends on that scientific Latin name, what exactly the species
Yeah.
is.
And honestly, I've never heard of a Buckeye before, so I'm sorry.
The next question was from Stephen L. Cantor. Please explain the origin of the number 54 and 58. Does that refer to chromosome locations?
That does not refer to chromosome locations. Instead, it refers to transformation events. So when we are generating these transgenic embryos and inducing shoots, we get hundreds and hundreds of them from each transformation. And so we need to screen them, and we have to label each one and track each one through the whole tissue culture and molecular process. It is impossible to keep track of those things with identifiers other than numbers because we have like individual letters and numbers. So it has nothing to do where the gene is inserted into the genome. It's just that is Darling 54. That is the 54th event that we've tested, and then we screen through.
Okay, thank you. Next question from Chrissy Vaughn. I have two hybrids that I purchased. They are an American Chinese hybrid, but I also have what I believe is an American tree that I grew from a nut. When these three trees pollinate together, would it be bad to grow future trees from those nuts? They have not yet produced any as they are only 3 and 4 years old.
Oh no, there's nothing wrong with hybrids, crossing them for your own purposes. Um, it's great that you have three trees, and the offspring will just be complex hybrids between American, Chinese, whatever their background genetics, and it'll just be kind of a random uh, randomization. There's no reason not to allow them to cross-pollinate with each other. In fact, if you don't, they won't produce nuts.
All right, that's very cool. Next question is from Jillian Lee. Do the plantings that you guys are doing count towards New York's initiative to plant 25 million trees, or is there a limitation because of the regulatory requirements?
Yeah, so there's a limitation due to regulatory. So everything that we plant currently, because we have not been granted the ability to plant outside of permitted locations by the USDA, everything that we plant is on a permitted site. So everything is mapped. We track survival for every individual at every location, and it all has to be mapped both virtually and in person. Um, Everything needs to be fenced in. We're not allowed to let the trees flower naturally. We have to pluck the flowers off, prevent spread of any kind. So everything is in highly controlled field settings at this point. Um, so even if we were counted towards the 25 million trees that New York wants to plant, there's no way that we would be able to fence the amount of area for that or produce that many chestnut trees that quickly.
We have a few more questions. Diane Stikowski, we have Dunstan chestnut trees planted. Are they a hybrid and blight resistant? And also, what is a mother tree? And mother is in quotations.
Uh, Sorry, say that type of tree again.
Uh,
Tungsten?
Dunstan, spelled
Oh, Dunstan.
Dunstan.
Yep. Yeah, so Dunstan is a complex hybrid. I don't know its levels of resistance. We don't grow them, but they're one of the most common ones that you can buy from orchard nurseries, those types of places. They're supposed to make good nuts. I don't know the backgrounds, the genetic backgrounds of those. Mother trees is a term that we use for are cross-pollination. So we collect all of the pollen from male flowers, and when we apply that pollen to a mother tree, we're just applying it to a tree that has female flowers out in the field that hasn't been pollinated yet. So it's just a delineation term. You could just say crossing them with other trees, but it just delineates that we're using the male pollen to put onto the mother tree.
Um, from Alan Hersker, thank you for your presentation and this work. I have two American chestnuts that I got from Fedco in Maine. They are, as I'm sure you know, vehemently against GE efforts, including those around American chestnut. Can you explain why they are so resistant? And I'm assuming this is just a general question, and why would some people be
Yeah.
resistant to genetic engineering efforts?
Well, I mean, there's a whole slew of reasons, and There have been tons of bad publicity around genetic engineering, mainly because of large agrobiotech companies such as like Monsanto, all of that stuff, where anytime a farmer accidentally produces some of their seed because some other farmer's pollen got onto their corn, they sue them for the product, that type of stuff. There's a huge, you know, reason that some people would be in outrage over those things. But it's more like what's your intended use of this? It's not like we are intending to uh, mass produce these trees and then only plant them where people pay us a bunch of money to. Our whole point is to restore the species, and everything has a mixing of genes, whether it's just crossing two individuals, two different species. And instead of just randomizing it, we're just doing a targeted effect. And I can't really speak to other people's motivations as to why they wouldn't get behind this effort as opposed to something else, but it really is something specific that it's really hard to, without specific reasons, address them, you know?
Yeah. And then for our last question, Heather Lee, are the 54 and 58 the same? I heard about the mislabeling. Can you please explain what happened? And I
Yeah.
think you kind of covered that, but can you just go over it one more time?
Yeah, no problem. So 54 and 58, they're just two different events that were screened around the same time. And what happened on that specific instance was uh, when we're tracking all of that information and producing pollen in the highlight growth chambers, two labels got mixed up between the Darling
Mm.
54 and Darling 58. So the difference between those two things is just where the oxo is in the genome. Both only have one copy, both are resistant to the blight. We just mixed up the labels. So when we did our USDA and FDA and EPA petitions, we described our trees as Darling 58s, when in actuality, most of them were Darling 54s. So all we had to do is go back to the USDA and the EPA and the FDA and basically just edit our petitions
Mhm.
to have a name change correction, basically. So we resubmitted all of those things, and all of the data that we have done is still viable. It's just we accidentally labeled it wrong.
Gotcha.
And that was like 10 years ago. So,
Yeah, things do happen. I
yeah.
did see in the chat someone followed up uh, that they read that the buckeye tree is indeed the same species as the horse chestnut. So that was from Kevin.
Yep.
My answer helped answer the first question. And sorry, one more question popped up. I thought it was really interesting from Stephen Cantor. What are the oldest transgenic trees that you have been growing now, and how large are they?
So the oldest transgenic trees that we have uh, are from events that are not the ones that we're going through the regulatory process with. So we have some Darling 4 trees, and
Mhm.
those are different constructs, but those trees are much older. They're like, I honestly cannot remember the year that they were planted, but they have to be 20, 25 feet tall. But the problem is we're not allowed to let them flower and we're not allowed to let them spread pollen. So we have to coppice them once they get too big because we can't go up and pluck every single flower off of every tree. Our darling trees, our oldest trees were planted in 2016 from our first crosses. So they are approximately 9 years old at this point. They've been in the field maybe 8 years, and they are probably 10, 12 feet tall, something like that.
Very cool. And I will say, to wrap this up, I see Alan Nichols in the chat said the New York chapter of the American Chestnut Foundation is supplying pure wild type American chestnuts to people who want nuts to plant. So that could be maybe something of interest. But other
Yeah, you
than
got to reach
that,
out to Alan. He is great about getting nuts to people who want them. And thanks for being here, Alan.
Awesome. Well, thank you. I know we ran a little bit over, but thank you so much, Dakota. Much appreciated. This was a really robust presentation. And I might go back and rewatch it once the recording's out to catch some of it because it was very, very chock full of information. So thank you so much for your
I'm
time and
sorry if it
expertise.
was too much.
No, it was great. It was awesome. So, and I'm seeing lots of kudos in the chat. So, lots of excitement about maybe getting to experience more of the chestnuts that our ancestors got to appreciate in the past, but in the future now. So, thank you for all your hard work.
Well, thank you for having me. I hope you guys have a great evening.
All right, you too.